plasma membrane is an incredibly important structure that regulates what gets into and out of the cell. It is a selectively permeable membrane. And what that means, selective, if you'll think back to talking about molecular interactions, you're aware of the term selectivity and that idea that it only lets, or in the case of selectivity, a protein only binds certain molecules. In the case of the membrane, it also means the same thing. Only specific molecules are allowed to pass through. The function of the plasma membrane are to physically isolate the internal portion of the cell. It regulates exchange with the internal environment. It's sensitive to environmental changes, provides structural support, and so forth. Importantly, when we talk about homeostasis, we talk about homeostasis in the person as a whole, of course, but we also need to remember that cells themselves are living organisms, and living organisms, or let me correct myself, humans are living organisms, bacteria are living organisms, and bacteria are cells. In our body, cells are alive. Each individual cell is alive, and it functions independently while also coordinating and working together with other cells. And so the internal environment of the cell is the ICF, as well as the environment in the nucleus and so forth. And so we have to apply the principles of homeostasis to a single cell as well. The internal environment is relatively stable within the cell. Changes do in fact occur, and those changes often are used as signals to change the function of the cell, but homeostasis still applies in this condition, and the plasma membrane is incredibly important for maintaining the internal environment of the cell, or to help with that process of homeostasis by controlling what actually gets into the environment as to what actually can change that environment. If we take a closer look at the plasma membrane, here's a lovely diagram of the plasma membrane. Let me identify the key components. Most of the plasma membrane is made up of the phospholipid, and as we can see here, that phospholipid is usually illustrated with a circle that represents the phospholipid head. Phospholipid heads are hydrophilic. They love water, and so they face the open water. And then we have these tail regions, which are fatty acid tails. Those are hydrophobic, and they tuck themselves away from the water on the internal layer of the cell. We have a phospholipid head facing the internal environment. So if we were to look at the interstitial fluid is down here, whereas the extracellular fluid is up here. And so in both cases, fluid being the key component, water is a very important part of that. Then we have our proteins, which is probably the next most important part of the cell membrane. We have proteins that span the entire membrane, such as this little corkscrew protein here. Goes all the way through that phospholipid membrane. We have an internal portion and an external portion. So we have the intracellular side, which is the side that's in this one. This is the intracellular side. We have the extracellular side outside of the cell. These little structures right here, these are carbohydrates. These are essentially sugars, and we have these carbohydrate chains, which makes this particular protein a glycoprotein, glyco meaning sugar. And you'll notice that we have a number of different carbohydrates attached to various different structures within the cell. We also see other proteins, the proteins that pass completely through the membrane. We have two different terms for them. These are basically transmembrane proteins. They're also known as integral proteins, and they cross this portion of the plasma membrane here. That doesn't show up very good. Let me show this portion here. And so the side that interfaces with the phospholipid, the fatty acid, this portion of the transmembrane, this part right here, the transmembrane protein, is hydrophobic, whereas the part that interfaces with this one will work, with the external side, the extracellular fluid or the intracellular fluid, in those cases, that region is hydrophilic. And so by hydrophobic and hydrophilic, I'm referring to the amino acids that interact with the environment. Another type of protein that we commonly see is a peripheral protein, as shown here. These are not entirely embedded. They don't cross the membrane completely. We have peripheral proteins in the inside. We have peripheral proteins in the outside. And you'll notice that they are embedded just a little bit, but they're not completely in the cell membrane, not buried in it the way that transmembrane proteins are. Then we also have our surface proteins. These ones are just touching, kind of riding along on top or on bottom of the phospholipid layer. Often these are referred to as lipid anchor proteins because there's usually a lipid that kind of keeps it close to the surface. And so those lipid anchor proteins will hold on to the phospholipid membrane. You'll notice that we also have the cytoskeleton on the inside of the cell. In addition to glycoproteins, we also have lipoproteins, proteins with lipids attached, and this is essentially the basic structure of the cell. The last thing I want to mention is the cholesterol. Cholesterol is shown here. I'm coloring it in white, and notice it tucks itself in between the phospholipids, specifically the fatty acid tails, and cholesterol's job is to stabilize the membrane. It essentially makes sure the membrane is fluid, but not so fluid that it's going to melt. So it's fluid yet solid. I know that doesn't make any sense, but it's kind of this in-between substance. I sometimes think of it like butter that you pull out of the refrigerator. You know, you've got a nice firm cube. That probably is not the greatest analogy, but it's what I've got for you. Here's a closer look at the phospholipid. So this phospholipid head is essentially a glycerol molecule, this part right here, attached to different chemical structures that give this molecule its property. The head is soluble in water. So if we were to clip that off, it would dissolve in water just fine. And so it's definitely hydrophilic. It loves water. The tail are these structures here. These are fatty acids, and you'll remember we talked about fatty acids and what those are, and those are hydrophobic. Some of the fatty acids are saturated fats, some are unsaturated fats, and the unsaturated fats create the double bond, and those unsaturated fats create a kink. The more saturation we have, the more those phospholipids are, the tighter they can pack together. Unsaturated fats, they pack more loosely, and this dictates how solid basically the phospholipid membrane is. The phospholipid membrane is what we call a fluid mosaic. This particular slide walks you through the peripheral proteins. Some other terms, I'm going to define these terms in a minute, but this, I'll just let you go ahead and read through this because I kind of got ahead of myself and talked about all of them up here. And so I'll just point out this side, make sure you read through this and look at this. Same thing with the integral proteins and transmembrane proteins. One thing I want to note about the integral proteins is that many of our sensors are integral proteins. Here we actually see a channel, so many of our transporters are channels, are integral proteins. Transporters that are not channels are integral proteins in many situations, and so they're very, very important in terms of function. Many of them are attached to the cytoskeleton, which anchors them firmly in place so that these particular proteins don't necessarily drift. The fluid mosaic model, what these phospholipids do is they kind of bob up and down. They maybe not go too far, but they're going to move around a little bit. And anything that is not firmly attached is going to also float around. So many of the surface proteins, for example, will float around in the ocean of phospholipids. But if a cell protein is anchored to the cytoplasm, it's not going to move. I'm sorry, cytoskeleton, it's not going to move. Here's the definitions I promised you. Glycocalx is the general term for it. Honestly, I don't actually ask you to know that term for the exam, but we have the most common, the ones that I likely will ask about or possibly at least reference on a future date are glycoproteins and our glycolipids and our lipoproteins. It's not on my list. Let me put it on my list. Lipoproteins. Proteins with lipids attached. Why it's not on my list, I don't know. I probably just forgot about it. It's kind of just this relationship. So I know it's a little bit like proteoglycans are carbohydrates with proteins attached, whereas glycoproteins are proteins with carbohydrates attached. So it's just kind of a relative abundance. You know, is the carbohydrate the bigger portion, or is the protein the bigger portion? So proteoglycans are that carbohydrate. We do see proteoglycans. We will talk about proteoglycans with bacteria. They actually make up a protective layer of bacteria, but that's really the only time I focus on them.